Welcome to the Wednesday afternoon lecture series. My name is Veronica Alvarez. I'm an investigator at NIAAA. And today it's really my honor, and I have learned to save those words for when they're really required, to introduce you the speaker of the lecture, Dr. Bernardo Sabatini. Bernardo Sabatini obtained a PhD from the Department of Neurobiology at Harvard Medical School and his MD degree from Harvard and MIT Joint Program in Health Sciences and Technology, not so long ago, 1999. Then following a family tradition, actually, Bernardo has dedicated his career to research and work in the laboratory. He did a postdoctoral fellowship in the laboratory of Carlos Rabora at Cold Spring Harbor Laboratories in New York. And after that, he joined the faculty in the Department of Neurobiology at Harvard Medical School in 2001 and has remained there since then. Bernardo has received numerous awards and honors during his career, and I'm going to start for some of the latest. In 2008, he was named an investigator of the Howard Hughes Medical Institute. In 2010, he was named Takeda Professor of Neurobiology at Harvard Medical School. Before that, for instance, he had received the Young Investigator Award from the Society of Neuroscience in 2008, McKnight Technology Award, Innovation Award. He has been a McKnight and Sears Fellow that funded his lab for a long time when he started at Harvard. So his laboratory focused on understanding the function and regulation of synapses in the mammalian brain, with particular interest in how the function of synapses is perturbed during disease. Some of them are autism, Parkinson, and Alzheimer that research projects have been focusing on lately. And I have to say that I have had the honor to be a postdoctoral fellow in Bernardo's lab and receive training there. And I think that beyond besides of his very impressive CV, it actually doesn't make justice to the really amazing investigator and researcher he is. He's extremely creative, and so working with him in science is actually a lot of fun. And I think another quality of Bernardo's, beyond of the usual, smart, hardworking, all those things that we all know are required for science, I think Bernardo is extremely brave. And I've learned, I have been inspired by this while I was working with him. I think you sometimes need to be brave and take risks to tackle these important and meaningful questions in science, and I think Bernardo is doing that really well. And examples of these, of his creativity and his lack of fear, is the amount of new technologies, both optical and chemicals, that are being developed in his lab in order to further study the function of synapses. So he's going to tell us about self-stimulation, how the brain wires itself, and surprising new ways that it turns itself off. Okay, sorry about that. So welcome. Thank you, Bernardo.
Okay, thank you, Veronica. I'm really honored to have been the subject of that introduction. And what Veronica didn't tell you is that she was actually the founding member of my lab. She arrived at Harvard about 2 months before I did and was the first person there, obviously, and really helped me get things going. And so a lot of the success that my laboratory has had is really due to people like Veronica, who are very pioneering and brave to join my lab when I was completely unknown. So thank you. So I'm going to tell you about, I'm going to push these away. I'm going to tell you about a project that was just completed in the lab. And is about to appear online. Hopefully too many of you have not read it yet, so it won't be boring for you. That's the first part. I think you'll understand the title in a minute. And then if I have time, I will get to some surprising new results that are totally unpublished and really unfinished, and that's the second part here about how the brain turns itself off. But I may not get to it. We'll see how we have time. So this is the animal that we study in the lab. This is a laboratory mouse shown here on the left. This is obviously a new mother. She has a bunch of pups there, a litter of pups. And one of the things we try to understand in the laboratory is the remarkable transition that happens between this stage of development and this stage of development. So just after birth, these pups are very limited in their behavioral repertoire. Their locomotor ability, both gross locomotor ability and fine motor skills, are incredibly poor. There's very little that they can do. Their sensory systems are not yet fully developed. And essentially at this stage, all they can really do is find their mother, suckle, and grow and continue to develop. But within a few weeks after birth, these animals become fully autonomous creatures that in the wild are able to forage over approximately a square kilometer. They're able to remember their way through their environment. They're able to associate sensory cues with the presence of rewards like food, water, or mates, or of aversive things like the presence of predators. And so we want to understand what occurs in the brain that allows this remarkable developmental maturation in just a few weeks. And so in particular, we're interested in these rapid progressions that are shown here, the rapid increase in locomotor ability, in navigational ability, and in particular, this ability to form associations between stimuli and rewards. And when we think about these kinds of problems, it's important to differentiate between two kinds of plasticity that can occur in the brain. So one is the changes in the brain itself that allow it to become an organ that can learn, right? So that's really a developmental question. What changes from here to here that this animal is now capable of doing quite sophisticated learning? That's one problem that we're interested in. As I said, that's really a developmental problem. That's mostly what I'll tell you about today. And secondly, related question is what actually changes in the brain itself when an animal acutely learns. So when this animal learns that this kind of leaf tastes particularly good and it's something that should seek out, what has happened in his brain? And that's a much more acute form of plasticity that's thought to happen at the level of changes in how cells talk to each other, how synapses act. Now, the part of the brain I'm going to tell you about today is the basal ganglia. And this is because the basal ganglia is necessary for many fundamental behaviors seen in mammals and in humans as well. And so, for example, the proper function of the basal ganglia is necessary to generate purposeful movements, and is also necessary for many forms of reward-based learning, reinforcement learning. And this is made quite clear by diseases of the basal ganglia in humans. So, for example, Parkinson's disorder and Huntington's disorder, two neurodegenerative and quite devastating disorders, are both diseases of the basal ganglia. Furthermore, drug addiction, many forms of drug addiction, of drugs of abuse, the mechanism of action of these drugs is actually by changing the activity of neurons within the basal ganglia. And so most drugs of abuse act by increasing the level of dopamine in one part of the basal ganglia of the striatum. And of course, that has very profound effects on human behavior and can really co-opt the whole behavioral repertoire of a person, drive them into intense drug-seeking behavior. There's emerging evidence that many other diseases, including obsessive-compulsive disorder, Tourette's syndrome, ADHD, and other neuropsychiatric disorders, also have components of basal ganglia dysfunction. So just to get everybody on the same page, I'm going to spend a little bit of time outlining the circuitry of the basal ganglia. And for those of you in the audience that are real experts in basal ganglia or that come from a primate physiology background, please understand that I'm going to show you a very simplified mouse-centric view of the basal ganglia. So the basal ganglia consists of two interlocking loops. So there's a projection from cortex, from many areas of cortex, into the striatum. This is an excitatory projection. These are cells that release glutamate and can stimulate the striatum. From the striatum, there's a projection of inhibitory neurons that goes to the substantia nigra reticulata. So this is an inhibitory projection that releases GABA. In turn, then, there's an inhibitory projection back to the thalamus, and then an excitatory projection back to cortex and striatum. So if you look at this loop, there are several excitatory projections and two inhibitory projections, and so the net sign of this loop is one of excitation. This is called the direct pathway, and it's thought that activity of the cortex acts through this loop to reinforce activity of cortex, so it's really a positive feedback loop. In parallel, there's a second pathway called the indirect pathway, in which there's an extra synapse. So the striatum, again, we have inhibitory neurons that make synapses into the globus pallidus, And then the globus pallidus sends a second inhibitory neuron to the substantia nigra reticulata. And so now in this case, we have 3 inhibitory synapses within the loop, and therefore the net sign of this is inhibition, and it's thought that the activity of the indirect pathway suppresses activity in cortex. This is a negative feedback loop. And this division between direct and indirect pathway is going to be something that we will pay attention to throughout the whole talk. Okay, so here's another view of the basal ganglia cortical circuitry. And what we thought was interesting about the development of this circuit is that here you have a circuit with many nuclei, many components in it, some of which are excitatory and some of which are inhibitory. And so it was unknown in the mammalian brain how such a circuit would develop and wire itself. What was the role of activity in these kinds of recurrent loops in stabilizing the circuitry and helping it form? And we know that a lot of the maturation of basal ganglia circuitry actually happens after birth, for example, in a mouse in the 2nd week of life. And so it's quite likely to believe that the experience of the animal, its sensory environment, and the patterns of activity in the brain in this postnatal period would actually play a role in governing the formation of this circuit. So now let's spend a little bit more time on these 2 pathways, the direct and indirect pathway, because the division is very important. So it turns out that both the direct and indirect pathway, these 2 pathways of information flowing through the basal ganglia, are formed by a class of neuron called the medium spiny neurons. Some people call these spiny projection neurons. I've just gotten used to calling them medium spiny neurons. These are the principal cells of the striatum. They form the output, and they're really a curious cell type because they're inhibitory, yet they send long-range projections, and that breaks one of the general rules in the brain, which is that the long-range projections are usually formed by glutamatergic excitatory neurons. Okay? As you'll see in a minute, these cells are also covered with a high density of what are called dendritic spines. And these are these peculiar mushroom-shaped specializations at which excitatory synapses are formed. It's also atypical for inhibitory neurons to have dendritic spines. So this is another one of these rules that's broken by this type of cell in the brain. There are 3 or 4 examples of cells like this in the brain that are GABAergic projection neurons that have spines. So now it turns out the direct and indirect pathway medium spiny neurons pretty much look the same within the striatum. You can't really tell them apart, but there are fundamental differences between them. One of which is an anatomical difference that I already mentioned. The direct pathway neurons project to the substantia nigra reticulata, whereas the indirect pathway neurons project to the globus pallidus. There are also molecular differences. These cells express the type 1 dopamine receptor, whereas these cells express the type 2 dopamine receptor. And that distinction is fundamentally important because these 2 types of receptors differentially signal to intracellular pathways. One of them activates PKA and the other one inhibits PKA. So there's a push-pull type arrangement there. They differ in other molecular respects. These guys express the peptide substance P as well as the opioid dynorphin, whereas these express the opioid enkephalin. What that difference really means and how it changes the function of the circuit is something unclear, but something that we're interested in. And furthermore, as proposed in very classic models of basal ganglia function, this direct pathway medium spiny neuron, when it's active, promotes movement of the animal, it promotes locomotion, whereas this one inhibits movement. And it's thought that Parkinson's disease, that one of the main causes of disease is an imbalance between activity of these 2 pathways, such that indirect pathway activity is favored in Parkinson's disease, and that explains some of the difficulty of people with Parkinson's in initiating movements. Now, another feature of the basal ganglia circuitry that we haven't touched on yet is that this kind of box and arrow diagram is actually quite deceptive. It's not that all of cortex projects to all of striatum, projects to all of globus pallidus. There are really several interlocking subloops such that one part of cortex will project to downstream areas and another one will project to other downstream areas. And so we really need to think of this as a series of loops between all these structures. And so that's the question that really was very interesting to us, because how can this system wire itself? How can we have connections that are maintained across 4 or 5 nuclei, some excitatory and some inhibitory? How does the cell know where to project in the downstream area in order to close this loop? Okay? And that's really going to be the subject of today's topic. And we thought that this kind of loop is more easily formed via activity-dependent wiring rules, that somehow the loop can detect when it's correctly wired based on the patterns of activity that lie within it. And that's what I hope to convince you of today. I want to stress that this is fundamentally different than the best studied circuits within the brain in terms of development, which are sensory systems. And so, for example, in the eye, we know that of course, the organization of the retina is topographic. It reflects the organization of the world outside us. So for example, nearest neighboring cells within the retina are receiving information from nearest neighbor portions of the visual field. And that topographic arrangement is maintained through various stages within the processing system for the visual system. So for example, within the thalamus and the lateral geniculate nucleus and within the cortex. Furthermore, early in development, even before an animal is capable of seeing, even a mouse, even before the eye opens, it turns out there are patterns of activity, there are waves of activity that are spontaneously generated within these structures that can project information about nearest neighbor relationships forward. So for example, there are waves of activity that occur in the eye that are transmitted forward into the LGN, transmitted forward into the cortex, that are passing information about what cells are near each other and therefore listening to, well, seeing neighboring parts of the visual field. And so there's a lot of theories about the development of this visual system in which it's clear that these kinds of waves, coupled with some learning rules for stabilizing and withdrawing synapses, is sufficient to explain a lot of the topographic arrangement of the sensory system. And this is work that's appeared over many decades from people like Carla Schatz, Rachel Wong, Marla Feller, Marcus Meister, and many others. Now, quite interestingly, there's been recent work by Nick Trish and Dwight Burgles showing that there's a very similar phenomenon in the auditory system. So that starting in the inner ear, where there's a tonotopic arrangement, so that nearest frequency responding cells are close to each other, that there are also patterned activity, waves of activity that propagate through that structure that also transmit information about nearest neighbor relationships to downstream structures. So in this case, we have at least a model for how topography can be organized. And I think that's fundamentally different than this circuit. First of all, this circuit is not a direct sensory circuit. It's embedded within substructure of the brain, sub-cortical circuit. Secondly, it consists of mixed excitatory and inhibitory projections, and there are multiple nuclei. So how can we have this system wire itself? And that was the subject of these two people here, Zhenya Kozarovitsky, a postdoc in the lab, and R.P. Saunders, a graduate student in the lab that worked together on this project. And so they decided, in order to understand if activity in the circuit plays a role in its development, the best strategy was to do what says here. Let's silence either the direct or the indirect pathway and understand how these imbalances of perturbation alter the circuit. Now these guys did the work, but since I'm at NIH, I want to really take some time to thank Chip Gerfin, who's here. And so Chip's efforts as one of the co-PIs of the GenSat project, along with Nat Hines, has really enabled, I'd say, all of modern basal ganglia research in the mouse. And so Chip was one of the people that really pushed forward the development of back transgenic mice that allowed GFP or Cre recombinase to be labeled, to be expressed in particular subpopulations of the basal ganglia. It's thanks to his efforts that we not only have the mice, but we really have well-characterized mice that allow us to put proteins into very specific elements of the basal ganglia. And you'll see in a minute how we use that technology. So what I'm showing you here are images of fluorescence patterns in a reporter animal that expresses a fluorescent protein under the control of Cre recombinase. And on the left, we've crossed that reporter animal with another mouse that expresses Cre recombinase under control of the type 1 dopamine receptor, DRD1A. Okay, and remember I told you at the beginning that the type 1 dopamine receptor is selectively expressed in the direct pathway medium spiny neuron within the striatum. And so what you can see here is fluorescence within the striatum, and then this coalescence of axons that are projecting from the striatum into the substantia nigra reticulata. And so this is the tool that we'll use to turn on expression of proteins within the direct pathway. On the right is a similar image obtained by crossing the reporter animal with a second animal that expresses Cre recombinase under the control of the type 2 dopamine receptor. And remember, this now labels the indirect pathway. And you can see these very short axons that go from the striatum into the globus pallidus. And so we took these animals and crossed them with another animal. This is an animal that was made by Brad Lowell at the Beth Israel at Harvard, which is a conditional or floxed allele of this gene, SLC32A1. This gene name isn't really important. The point is that this gene encodes a protein known as VGAT, the vesicular GABA transporter. And this is the only protein known to take GABA from the cytoplasm and package it into synaptic vesicles. And so the idea is that this mouse now will lose expression of VGAT in cells that express Cre recombinase. And therefore, we can silence one pathway or the other pathway and see what the effects on development are. And so the D1 Cre VGAT flox animal will have a silenced direct pathway, whereas the other animal will have a silenced indirect pathway. It's important to keep in mind that we don't expect this to be toxic to the cells in any way. Where the cell should still be able to produce synaptic vesicles, they simply won't have GABA in them. So they just won't be able to output to the downstream nuclei. So here's an image of these mice. Turns out that these mice are quite sick, and they all die by about 20 days of age. We can keep them alive a little bit longer by culling the litters such that only the mutant mice are present with the mother, and that allows them to get better access to the mother and survive longer. And so here's an image of a mouse that has one copy of VGAT in the direct pathway compared to a litter mate that has 0 copies. Obviously, this animal is runty, and as I mentioned, is quite sick. The indirect pathway silenced animals are a little bit better off. They survive a little bit longer. They're a little bit runty as well. They're smaller in weight, but not as severely affected. Okay. Now, what's interesting is that these animals, despite being sick, already show the locomotor phenotypes that one would expect for perturbations of the direct and indirect pathway. So this, what I'm showing you here, is video tracking of the movement of these animals in an arena over an hour. And what you can see that at P14 days of age approximately, this animal here locomotes. This is a control. It has one copy of VGAT, whereas its sibling that has no copies of VGAT shows much reduced locomotor ability. And this is consistent with these models of classical basal ganglia function that were proposed actually by Gerfin and others many years ago, and were very recently proven to be true by acute manipulations of direct and indirect pathways by people like Lex Kravitz, who's actually coming here to the intramural program. He did that work as a postdoc in Anatol Kreitzer's lab. So the direct pathway silenced animal is quite poor in its locomotor ability, whereas the indirect pathway silenced animal, despite being sick and runty, shows greatly exaggerated locomotor ability. So again, consistent with this push-pull model. For the sake of today's talk, the importance here is that even at these young ages, P14 to P17, already the opposing organization of the basal ganglia is in place, such that the direct pathway has one behavioral phenotype, whereas the indirect pathway silenced animal has the opposite behavioral phenotype. So we've done a lot of analysis of these animals. I'm going to skip many details, but a couple of controls that I just want to show you are that the number of cells of the direct and indirect pathway in each one of these animals is unperturbed. So it's not as if silencing the direct pathway makes these cells die. There are just as many direct pathway medium spiny neurons there as in a control animal, and same for the indirect pathway. Furthermore, the axons of these cells, and this was surprising to us, are perfectly fine in reaching their targets. So you can see here fluorescence filling the substantia nigra reticulata in a null animal compared to a control, or the globus pallidus in a null animal and a control. And we've done more refined analysis of this to convince ourselves that the fine structure is okay as well. But for the point of this talk, just keep in mind that cell survival is fine and downstream targeting of nuclei is fine as well. So obviously the animals are sick. There's some major perturbation of circuitry. And so since the output of the system seemed to be correct, we decided to focus instead on looking at the input of the medium spiny neurons. So this is an image of a medium spiny neuron that's now been filled with a fluorescent molecule through a whole cell recording pipette. So we can record electrical currents through the pipette, and we can image the structure of the cell based on the fluorescence. You can see it's a compact cell. It's about 200 micron long dendrites that are covered, as I mentioned, this high density of dendritic spines. And what we were surprised, and so here's a blow-up view of a dendrite. You can see these spines on them, these lollipop-shaped protrusions. And in this cell, as in many other types of cell in the brain, there's a very close correspondence between the number of glutamatergic synapses and the density of these spines. So it turns out that most of these spines house one glutamatergic synapse. Okay, so you can count spines as an indirect proxy to count the number of glutamatergic synapses. And what we were surprised to find is that silencing the output of this cell has tremendous effects on its input, on the number of synapses that it receives. Okay? So here's an image of a direct pathway medium spiny neuron. Here's a blow-up of a spiny section of a dendrite from a control animal, and here's from a silenced animal in which 2 copies of VGAT have been lost. And what I hope you can appreciate is that this normal density of dendritic spines is greatly reduced in this animal. In fact, it's lost about half of its dendritic spines. Furthermore, if we look at the electrical recording of the cell, we can see these very short bursts of current flow, which correspond to the spontaneous activation of a single synapse. And by counting how often these occur, we can get another measure of how many glutamatergic synapses are present on the cell. And I hope you can appreciate from this example that also the number of glutamatergic synapses measured by this electrophysiological assay is greatly reduced in this cell. And so from this kind of data, what we've learned is that silencing the direct pathway decreases the density of dendritic spines and the frequency of miniature EPSCs, this is electrical analysis of currents, onto these cells. Okay? Now the surprise came when we analyzed the effects of silencing the indirect pathway. And so this is an indirect pathway medium spiny neuron analyzed in the same way. And I hope that now you can appreciate that the silenced neuron has many more spines than the control neuron. Okay? So remember that, so silencing the indirect pathway now enhances the density of dendritic spines. Furthermore, electrophysiologically, we see an enhancement of the frequency of these events, both of which lead us to the conclusion that silencing the indirect pathway increases the density of spines and the activation of synapses on these cells. So the opposite effect that we saw before. And so here's the quantification of that data. So let's start up here. If we silence the direct pathway and look at direct pathway neurons, we see less electrophysiological correlates of synapses and less morphological correlates of synapses, less spines. Conversely, if we silence the indirect pathway, we gain electrophysiological correlates of synapses and gain morphological correlates of synapses. And so at this point, there are really two possibilities that we need to consider. One is that maybe this gene that we've knocked out in its protein product, VGAT, has opposing functions in these two cell populations. Somehow, this gene, even though what it does is package GABA into vesicles, acts to reinforce synapses in direct pathways so that when we lose them, when we lose the gene, we lose the synapses. And in this case, acts to destabilize the synapses so that when we lose the gene, we increase synapses. That's one possibility. The second possibility, of course, is that by silencing these two pathways, we have opposing effects on the circuitry, and we're really revealing some kind of activity-dependent learning rule within the system. And I hope to convince you the second one is true. Now, the first piece of data that we had that the second model was likely true was obtained by looking at the opposite pathway. So when we silence the direct pathway, what happens to the indirect pathway, which is genotypically normal? We haven't done anything to it. And what you see is that the indirect pathway also loses electrophysiological correlates of synapses and morphological correlates of synapses. And when we silence the indirect pathway, we saw the opposite effect. Silencing the indirect pathway increased the number of synapses onto direct pathway medium spiny neurons and the density of spines, even though those cells were genotypically normal. And so I think this is best explained by a circuit effect as opposed to a cell autonomous effect of that gene. And so what we're really saying is that when we silence the direct pathway, we lose this inhibitory projection. Because of that, this structure is overactive. Because of that, this is over-inhibited, and therefore drive in the circuit is reduced. And so we would predict that silencing this would reduce activity in the whole circuit, and if this circuit pays attention to its own activity to drive synaptogenesis, it would therefore lose synapses. Conversely, silencing the indirect pathway would have the opposite effect. It would boost activity in the circuit and possibly promote the formation of synapses. And so we wanted to test that model directly, which means that we wanted to test the model directly, and we also wanted to do it in a temporally restricted manner, as opposed to restricting the activity of these cells from birth, as I showed you in the previous slides. And so to do that, we need to have a way of altering the activity of genetically defined neurons during a defined temporal time window after birth. And so we turned to a tool called HM4D. This was a G protein coupled receptor that was developed by Brian Roth. And what he did is he took the muscarinic cholinergic receptor, which normally couples to the activation of potassium channels and silences neurons, and he mutated that receptor such that it no longer binds acetylcholine, but instead can be activated by a small molecule, a derivative of clozapine N-oxide, or CNO. And so the point now is that if we can take this protein and express it in particular cells in the brain, and then administer CNO to the animal, we can selectively reduce the activity of those tagged neurons. Okay? So we took this from Brian Roth, and we packaged it into a Cre-dependent recombinant adeno-associated virus, which we could use to infect the brain and therefore direct the expression of HM4D only in cells that express Cre. So let me show you some data that works. This is an image of a striatum of an animal that's been infected with this HM4D containing virus. In this case, the HM4D is fused to a red fluorophore called M. cherry, and you can see the expression of the protein within the cell. And just in this anecdote here, you can see that we can take a cell that spikes robustly and inhibit its spiking by the application of CNO. And so the way we quantified if the system really works is to use a combination of two viruses. And so what we did is inject into the brain of, in this case, a direct pathway labeled animal, a D1 Cree animal, two viruses, one that encodes the HM4D M. cherry that I just told you about, and the second one that encodes, also in a Cree-dependent manner, another protein called channel rhodopsin. And so this is an ion channel which is gated by blue light that, of course, Karl Diezeroff has introduced to neuroscience and made a useful tool. And so what we see is that neurons that express channel rhodopsin we can turn on with light. And so essentially here below you see that if we record from a medium spiny neuron, when we give a flash of blue light, we can increase the activity of the cell. And then we can wash in the CNO and reduce the activity of the cell. And now so we've shown essentially that a constant amount of drive to the cell provided by channel rhodopsin is less effective at spiking the cell. So the CNO is able to inhibit the activity of cells, and we can recover that with more laser power. We've done this kind of analysis for the D1 Cree animal, it works there. For the D2 Cree animal, it works there as well as for an animal that I'll tell you about in a minute called the RPB4 Cree. Just store away that this approach works there as well. So we can reduce the spiking of these cells by around 30 to 50% using this trick of HM4D plus CNO. And so we looked at two manipulations. First, we injected a very small amount of adenovirus into one side of the brain in the striatum. And so we got very sparse infection, so there were very few of these red HM4D expressing cells within the striatum. And then we delivered CNO to this animal from P8 to P14 during this time of maturation of the striatum and asked what happens. And essentially what happened is nothing. So individual cells whose activity had been knocked down during this time of P8 to P15 had no perturbation of dendritic spines or of miniature EPSCs. I'm not going to show you that data in the interest of time. And it didn't matter if we inhibited the direct or the indirect pathway. And so this kind of data says that sparsely inhibiting medium spiny neurons has no effect on synapse number, and so that it's not really the activity of a single cell that matters in determining its innervation, but likely the activity of the circuit. And so to prove that it's the activity of the circuit that matters, we did the converse experiment in which we now bilaterally injected large amounts of AAV so that we could infect most of the direct pathway or indirect pathway medium spiny neurons, depending on the example. And then once again delivered CNO from this period from P8 to P15. And what you can see is that now with this widespread infection, widespread perturbation of circuit activity, we can now cause direct pathway medium spiny neurons to lose half their spines and half their synapses, and we can cause the indirect pathway neurons to double their spines and double their synapses. And so this is now telling us that activity in this now more restricted developmental window in the second postnatal week is what acts to govern the formation of the striatal circuit. And so here's just the quantification of that data. We lose about half the spines in the direct pathway when we inhibit activity during this time window, and we gain a lot of spines in the indirect pathway. There's a lot of noise in this data because the basal density of spines and synapses in these cells is actually quite low. So in contrast to what I showed you before, widespread inhibition of spiking in medium spiny neurons with this pharmacogenetic tool, HM4D plus CNO, reveals these same pathway-specific effects on spine number and synapse density. So let me summarize some of the data that I've shown you here before I move on to further analysis of this model. So what I've shown you is that genetic inhibition of striatal direct pathway medium spiny neurons causes a hypoinnervation of those same cells and of the striatum in general. On the other hand, genetic inhibition of the indirect pathway, there's a typo there, sorry, causes a hyperinnervation of striatal medium spiny neurons. So we get converse effects. Pharmacological manipulations of these two pathways during the second postnatal week causes the same effect. And so what we're proposing is that glutamate release in the striatum directly drives synaptogenesis. And that furthermore, it's the activity of these innervating axons that are the ones that are causing the synapse formation process to get triggered. And of course, since we're silencing the output of these cells, that's seeing effects on the inputs, this also suggests that activity is acting through the recurrent loop in order to drive the formation of this circuit. Okay, so there are two questions really embedded in that proposal. One is it really the ongoing activity of cortical axons in the striatum that drives synapse formation? Is it really the activity of these corticostriatal projection neurons that's driving synapse formation here? And so luckily here, we were able to use yet another tool that Chip Gerfin gave to us, which is this RPB4 Cre line. And so this is really quite a beautiful mouse that expresses Cre recombinase in a high percentage of corticostriatal projection neurons. These are deep layer corticostriatal projection neurons. And so here we've crossed this animal to, again, this red report allele, the TD tomato floxed animal, and what you can see is expression of the TD tomato of the red fluorophore in these deep layer corticostriatal projection neurons. And furthermore, if we look in the striatum, you can see dense innervation of the fibers of these cells. Okay, so now we have a tool by which we can manipulate specifically the cells that project from cortex into striatum. And it's important this line does not affect, for example, corticothalamic projection neurons. It really is specific to corticostriatal. Okay, so here's an image of another mouse. This is an RPB4 Cre mouse, no red reporter in this case, that's been crossed in with a second allele, a second back transgenic allele. This is GFP under the type 2 dopamine promoter. And so this allows us to visualize indirect pathway medium spine neurons in the striatum while manipulating the proteins expressed by the corticostriatal projection neurons. And so in this case, we've come and injected into cortex the same adeno-associated virus that carries the HM4DM Sherry fusion protein. And so if we blow up an image of striatum and now turn off the green channel, we can see this beautiful pattern of innervations of corticostriatal projection axons entering the striatum. And as you can see, we can hit a huge number of these axons. And so now we have a tool to directly manipulate the activity of these cells during development and ask what the effects are on indirect pathway or direct pathway medium spine neurons. And so now what happened, consistent with our model, is that when we reduced the activity of cortical neurons, again during this narrow time window, we were able to reduce the innervation of both indirect pathway and direct pathway medium spine neurons, again cutting it down in about half. Furthermore, as expected, we also lost dendritic spines. And so this shows that the ongoing activity of cortical neurons, right, normally drives the process of synapse formation in the striatum, such that when we turn that down, we get less synapses in both pathways, consistent with our model. Okay, so now I want to take a slight detour. The second part of our model is that glutamate release is sufficient to create a new synapse. So is it true that exposure of a neuron to glutamate alone can make it form a new synapse? And this is a question that Hongbae Kwon, a postdoc in my lab, had previously addressed in cortex, in which he had shown quite beautifully that glutamate was sufficient to form a new synapse. And I'm going to show you a little bit of that data. It's published last year. And so Hongbae had been studying the process of postnatal synapse development in the cortex, which is equally impressive as in the striatum, in which from day 6 to day 21, there's a massive increase not only in the complexity of the dendrite, but also in the density of these dendritic spines and the number of glutamatergic synapses formed onto the cell. And what Hongbae was able to show is that glutamate can drive this process. And so the tool that we used for that is this molecule that's shown on the left. This is called MNI glutamate. And so glutamate is this portion here in which there's usually a carboxylic acid here. And this has been derivatized with this aromatic light-absorbing group. And when this group absorbs light, typically in the ultraviolet spectrum, it triggers a hydrolysis reaction at this bond, releasing glutamate and the inert caging group. Okay? So using this approach, we can now use brief pulses of light to release glutamate wherever we want. And we do this under 2-photon excitation. This allows us to image a cell, see a spine, for example, and deliver a pulse of glutamate to that spine that can elicit activation of glutamate receptors. And so Hongbae did that, but instead of targeting spines, he targeted regions of dendrite in young neurons that had no spines. And what he saw was really quite remarkable, which is that with repeated stimulation of a single site over 20 seconds, 40 pulses of glutamate delivered over 20 seconds, he was able to very quickly induce the growth of a new spine. Okay? And he did a lot of work, which I'm not going to show you now, to prove that these new spines are associated with new functional synapses. they have both of the dominant types of glutamate ionotropic receptors, AMPA and NMDA type receptors. And this process of new growth requires the activation of NMDA receptors, PKA, and MAP kinase. And this is a developmentally regulated process that occurs in young animals less than P20 days of age. So here's a schematic of that signaling pathway, very crudely outlined, in which glutamate activates NMDA receptors, and including a component of calcium released from the endoplasmic reticulum, we think activates adenylate cyclase, most likely calcium-activated adenylate cyclases, leading to activation of PKA through a mysterious pathway, also activation of MAP kinase, and then the growth of a new spine. Now, what intrigued us about this pathway for the striatum is that modulators of PKA are very prominent in the striatum. So, for example, dopamine is a modulator of PKA. In the direct pathway neurons, it turns on PKA expression, whereas in the indirect pathway, it shuts them down. And there's lots of hits in pathways or parts of this pathway that may be involved in manipulating synapse formation. So Hung-Bae had shown that before in cortex. Does this also apply in the striatum? And so we returned to the striatum and looked at this protocol. And so here on the left, you're seeing a section of the striatum of a D2 GFP animal. So GFP is expressed in indirect pathway medium spine neurons. This animal also carries 1 or 2 copies of this Cre-dependent red reporter. And then we very sparsely infected the striatum with an adeno-associated virus that encodes Cre recombinase in this case. So the very small number of medium spine neurons that were infected with the AAV will now express Cre, turn on the red fluorophore, and allow us to see their dendrite. And so in this way, what Genya was able to do is pick stretches of dendrite in which there were holes where there were no spines, and now deliver glutamate and grow a new spine. And indeed, the protocol in striatum worked just as well as it did in cortex, and she was able to, with about 50% efficiency, so 50% of the trials rendered a new spine, she could grow a spine in either direct pathway or indirect pathway medium spine neurons. And so this kind of data shows that glutamate release in the striatum is sufficient to drive synapse formation, and favors a model that we've been building up over the last few years, in which we think that it's really the activity of the axons as a source of glutamate that fundamentally remodels the circuit by driving such activity-dependent synapse formation. Okay, so I've been talking to you about this loop, and I've shown you some data on how the direct pathway and the indirect pathway can influence the synaptogenesis in the striatum in an activity-dopamine manner. Now, one thing that I've left off of this conversation so far, which is the subject of this last part of the talk, is dopamine itself. And so I mentioned that dopamine can modulate PKA. Now, of course, there's a major source of dopamine within the basal ganglia. This is substantia nigra pars compacta, illustrated here in purple, which sends dopaminergic projections essentially to all nuclei within the striatum, and in fact to a lot of the brain, but very densely innervates the striatum. Now, we know that dopamine release in the striatum is a fundamental part of reward reinforcement learning. Okay? And as I mentioned at the beginning, this is how drugs of abuse act. And so it would be natural to think that dopamine itself might play a role in regulating synaptogenesis in this structure. And indeed, in more preliminary data, this is what we find. And so here's an example of the tracing of the movement of an animal. This, in this case, a very young animal, about a P12 mouse. And you can see the P12 mouse barely moves around in the cage in an hour. And after we give it an agonist of the D1 receptor, SKF81297, turns on directly the D1 receptor, now this animal runs around like crazy. And this is a, we think, PKA-dependent process, because if we administer H89, a blocker of PKA, albeit not a great blocker of PKA, nevertheless a blocker of PKA, we can prevent this SKF-induced running of the animal. Now, so here's the data there. On average, we get about a 2-fold difference in the locomotion of these animals over an animal after giving SKF, and that's blocked by H89. What was really dramatic is that in that single hour of treatment with the D1 receptor agonist, these cells massively grew new spines, and I'm not going to show you the data, also formed new glutamatergic synapses. And so I think this difference is quite clear in a saline-treated animal versus an SKF-treated animal, and on average, we about doubled the density of dendritic spines in just a 1-hour exposure to a D1 receptor agonist. And as I said, this also occurred with the number of functional synapses. Okay, so to summarize this data, what I've shown you is that decreasing of spiking or elimination of GABA release in direct pathway medium spine neurons decreases spine density and functional glutamatergic synapses of both direct and indirect pathway medium spine neurons. And the indirect pathways have the opposite effects. Furthermore, decreasing the activity of corticostriatal projection neurons also decreases synapse number and spine density here. And the glutamate release is sufficient to drive synaptogenesis in the striatum. Furthermore, the D1 receptor activation promotes spine formation in a pKa-dependent manner. And so based on that, what we propose is that activity guides synapse formation in the basal ganglia. Furthermore, it acts in a positive feedback loop, so that manipulations that turn on the direct pathway and hence turn on the activity of cortex are self-reinforcing by driving more synaptogenesis within the striatum. And we think that these mechanisms allow for self-guided wiring of this loop through multiple stages. Essentially, as the loop randomly tries different connections, when it finds one that reinforces the propagation of activity through the whole loop, that drives more synapse formation and therefore self-reinforcing. Dopamine can accelerate this process in direct pathway medium spine neurons, potentially allowing for a context-dependent component to this regulation of the loop, such that, for example, when an animal performs a locomotor action that lets it find the mother and suckle, this would release dopamine and therefore reinforce that locomotor action. And so we propose that early in development, the direct pathway innervation occurs first and is enhanced when reward. This process is self-promoting versus recurrent activity, and that later the indirect pathway innervation follows and provides a break for the system. And we're working on more rigorously proving the second point. Okay, now in the last few minutes that remain, I want to switch topics a little bit. I've already introduced you to dopamine, and we know that dopamine acts as a long time scale neuromodulator, both of striatal function and of behavior. But there's still a lot of questions as to what exactly dopamine does in the striatum, and we were very much interested in this question, which is dopamine purely a regulator of plasticity? Does it just change the state of the striatum to allow growth or retraction of synapses? Or is it also a dynamic regulator of striatal activity on, let's say, the sub-second to second time scale? Now, it's well described by work of the group of Antonello Bonci and separately by the work of Tepper, and also has been long time proposed by David Sulzer at Columbia, that dopamine neurons also produce and release glutamate, okay? And so by releasing glutamate, we already have one mechanism by which dopaminergic neurons could quickly alter striatal activity. And so two postdocs in the lab, Nick Trish, with the help of Jun Ding, a second postdoc in the lab, decided to address this problem. And the way they addressed it is, again, using these modern tools. They took a dopamine transporter Cre driver animal, so this expresses Cre in dopaminergic neurons, and they infected that animal, again with an adeno-associated virus, but now in the substantia nigra compacta, and this virus carried channel rhodopsin, this blue light activated ion channel. They let this animal then return to its cage for 30 to 100 days, and later cut an acute brain slice and use blue light to stimulate these fibers that normally release dopamine and are coming from substantia nigra while recording the consequences on medium spine neurons postsynaptically. So here's an image of the brains of such animals. Here you can see a coronal slice showing substantia nigra compacta and VTA. In this case, Nick injected a virus that expresses GFP in a Cre conditional manner just to see what the efficiency and the specificity of labeling was. And you can see here on the right of a blow-up of that image, a stain towards tyrosine hydroxylase, one of the biosynthetic enzymes in the dopamine pathway, and then the expression of GFP. And you can see that he can really target the compacta section of this area, and that there's really all of the GFP is expressed in tyrosine hydroxylase positive neurons. I'm not showing the quantification here, but that's really true. Furthermore, if we then look in the striatum, we can now see the fibers of these axons shown here in green from the GFP densely innervating large portions of this dorsal striatum. And so what Nick first did is some carbon fiber amperometry to measure dopamine release within the striatum following a blue flash. And so here you can see schematized a short flash of blue light, I think it's a millisecond long, followed by a transient in dopamine measured by carbon fiber amperometry. And it turns out this transient has all of the properties that one would expect for true vesicular release of dopamine from these fibers. So it's blocked by a voltage-gated sodium channel antagonist necessary to fire action potentials. And it's blocked, importantly, by two drugs that we'll return to, reserpine and tetrabenazine, which are antagonists of the monoamine vesicular transporter. So this is the protein that's necessary for putting dopamine into vesicles. If you block that transporter, as expected, you lose dopamine release. And this really mimics a Parkinsonian state in which dopaminergic neurons are lost. Furthermore, if we use alpha-methyltyrosine, an antagonist of tyrosine hydroxylase, so of that enzyme in the biosynthetic pathway for dopamine, we also greatly diminish the release of dopamine consistent with depletion of dopamine from these cells. And so here's the quantification of that data. We don't need to go through it all, but the bottom line is that this looks like good vesicular release of dopamine from dopaminergic fibers. It's inhibited by antagonists of VMAT2. Tetrabenazine we can wash out and recover some of the action. It's inhibited by inhibitors of tyrosine hydroxylase, of voltage-gated calcium channels, voltage-gated sodium channels. We can recover this. But most importantly is what's shown here on the right, is that this is knocked down by about 2/3 by an agonist of the D2 receptor. And it's known that dopaminergic neurons actually express D2 receptors on their axons, and this acts as an autoreceptor to lower dopamine release. Okay? So all of this is just controls to say that we're getting good release of dopamine from these cells. So what happens now when we look at medium spiny neurons? And this is where we had a real surprise. So here's a recording from a direct pathway medium spiny neuron in an acute brain slice where we're locally illuminating with blue light in order to evoke dopamine release from these fibers. And so Nick has injected current in a step here to elicit robust firing of action potentials. And what you can see is that when he flashes the blue light, what we see is a pause in the activity of the medium spiny neurons. And this was really surprising because it was not expected from the actions of dopamine, nor of course from the actions of glutamate, which should increase the excitability of these cells. And so here's a quantification of that data. If we just look at the interspike interval before the blue light flash, right around the blue light flash or after, we can see that when the blue light flash comes, there's a slowing of the spike rate consistent with an increase in the interspike interval. And surprisingly, this change was independent of the action of dopamine receptors. So if we block dopamine receptors pharmacologically, we still got this increase. Instead, this pause was mediated by activation of GABA receptors, shown here in purple by the lack of effect in the presence of GABA A receptor antagonists. This effect is seen in both direct pathway medium spiny neurons and indirect pathway medium spiny neurons. So what we conclude from that is that stimulation of dopaminergic fibers rapidly inhibits the firing of striatal projection neurons. And so there are really two models we need to distinguish here. One is that these cells release three neurotransmitters. We know that they release dopamine. We've already been reported that they release glutamate. Maybe they release GABA too. So direct inhibition of these cells. And we can contrast that instead with a disynaptic indirect model in which what this cell does is only release dopamine and glutamate, but that these guys activate an intermediate neuron which then subsequently releases GABA, an indirect system. And so we turned to whole cell voltage clamp recordings in which we manipulated the potential of the cell to isolate either the glutamatergic current or the GABAergic current here in red, and then used pharmacology. And you can see here when we apply this compound, a blocker of GABA A receptors, we lose the GABA A receptor mediated inhibitory current shown here in red while preserving the glutamatergic current shown here in gray. Now if we add blockers of ionotropic glutamate receptors, we now eliminate the glutamatergic current. Now you can also do the experiment in the opposite way. If this were a disynaptic model, then the release of GABA would require the release of glutamate to stimulate that intermediate cell. And that's not what we see. Here in a direct pathway medium spiny neuron, you see that when we add the antagonist of the glutamate receptors, we lose the glutamatergic response, but the GABAergic response is preserved. And now we put on the antagonist of the GABA A receptors, both of them are gone. So this suggests that the release of GABA is independent of the action of glutamate, and I already showed you that it's independent of the action of dopamine. So this really favors the direct model. Another piece of evidence in favor of the direct model is if we look at the latency of the GABAergic current and the glutamatergic current, you can see that they're really identical. So if we consider the onset kinetics of the iPSC versus the EPSC, the time to half maximal activation or the time to peak, we can see that if anything, the iPSC precedes the EPSC a little bit. So the GABA activation comes a little bit before the glutamatergic. This is really an artifact of these currents being so much bigger than these currents that they emerge from the noise faster. But it's certainly not true that the glutamatergic comes first. Furthermore, we can really show these are independent by doing a lot of pharmacological experiments. No reason to go through all of these, but the EPSC is blocked by glutamate antagonists. It is untouched by GABA receptor antagonists. but it's reduced by this D2 receptor agonist consistent with it coming from the dopaminergic fibers. The iPSC is blocked by two different GABA A receptor antagonists, independent of GABA C receptors, independent of glutamate receptors, and once again sensitive to D2 receptors. It really looks again like direct release. I'm not showing you all of the things we've done, but we can really discount this model at this point and favor the direct release model. Now, in a sense, this was good news because we already knew how to eliminate GABA release from a neuron. I showed you that before. We have this vesicular GABA transporter conditional allele. We can lose that protein and lose GABA release. Okay? So we said, okay, VGAT is what packages GABA into vesicles. So let's cross the dopamine transporter Cree animal to the VGAT flox animal and generate a mouse that cannot release GABA from dopaminergic neurons. We can study the behavioral effects. So Nick went in to confirm that this was true. Again, in control, we can get the GABAergic current in red, the glutamatergic current in gray. And to our surprise, we found that the VGAT conditional animal has perfectly good GABA release. And this is really GABA A receptor activation. It's again blocked by the GABA A receptor antagonist, and the glutamatergic response is normal. So this was really quite puzzling. We went ahead and made the VGLUT2 conditional animal as well because we came up with some crazy theories as to how maybe the glutamate transporter is actually packaging GABA and whatever else. We made this animal, and what we found is that, of course, we abolished the glutamate response, so we can do this kind of experiment, but once again, the GABA response was still there. So now we have a situation in which the release of GABA from dopaminergic neurons is independent of the vesicular GABA transporter. And so we hypothesized, well, there's another transporter there. The monoamine transporter is present on these vesicles. It normally packages dopamine. This is the VMAT2, vesicular monoamine transporter 2 is what's expressed in these neurons. And we said, well, maybe that itself packages GABA. And in fact, that's what we found. We used 3 different antagonists of the vesicular monoamine transporter, and we abolished the GABA response. So here with reserpine, an irreversible antagonist, and we treated the animal before cutting the slice. We see the GABA response is gone. The glutamate response is there. Using tetrabenazine, this reversal antagonist, we can also block it, and yet a third antagonist, RO41284. So 3 different compounds, all of which target VMAT2, abolish GABA release without altering glutamate release. Okay? Now, I'll just quickly move through this because I'm really running long. So we said if dopaminergic neurons make GABA and use VMAT2 to package it, then we should be able to block VMAT2 pharmacologically and replace its action by putting VGAT into these cells. And so we made a virus that expresses VGAT in a Cree conditional manner, infected these cells, and then treated them with reserpine. And indeed, in that condition, we could now get mammoth GABA release from these cells that is now reserpine insensitive. Okay? So this suggests that dopaminergic neurons have everything they need to release GABA. They just need a transporter, and it can be VMAT2 or it can be VGAT. Furthermore, the latency of these responses were all still consistent with direct release, and it was all GABA sensitive and whatever else. So VMAT2 is necessary for GABA release, but it can be replaced by VGAT. So lastly, the last piece of data I'm going to show you is that if VMAT2 is really a GABA transporter, then it should be able to replace VGAT. Okay? So we took medium spiny neurons and generated an animal that lacked VGAT in the indirect pathway, and now infected these cells with both channel rhodopsin and a virus, and infected these cells with channel rhodopsin. And we can see that if we record from a neighboring neuron, we can now get light-evoked GABA release that's blocked by GABA A receptor antagonists. On the other hand, if we recombine out VGAT from the medium spine neurons, as I showed you in the first half of the talk, we can greatly abolish, if not fully eliminate, that GABA release from these cells. So this is GABA release that's dependent on VGAT. Now if we go in and infect these same cells with a virus that encodes VMAT2, we can at least partially rescue this GABA release. And there are many reasons why this should be a partial rescue, because you have to favor not full infectivity of the cells in order to get this to work correctly. So based on this, we say that VMAT2 can at least partially rescue GABA release in a typical GABAergic neuron that now lacks VGAT. And so based on all that, we come to the conclusion from a lot of genetics, optogenetics, and pharmacology experiments and viral experiments, we've shown that stimulation of dopaminergic fibers activates GABA receptors on medium spiny neurons that's sufficient to influence the spiking. Inhibition is via direct release of GABA from these terminals. It's independent of VGAT but requires VMAT2. And as I said, VMAT2 can rescue GABA release. And so this really leads us to conclude that VMAT2 is necessary and sufficient for vesicular GABA release, and that VMAT2 is most likely to be a vesicular GABA transporter acting in these cells. And so all of that, so what? You know, is this just a curiosity? I think it's really more than that. A few things that we draw from this is one, that dopaminergic neurons really are rapid modulators of striatal activity. In fact, we think they mediate an inhibition, a rapid inhibition of the striatum. And I think this needs to take into account and alter our models of reinforcement learning. Secondly, many monoaminergic neurons, including histaminergic, noradrenaline neuron, and serotonergic neurons, all rely on VMAT2 for the packaging of monoamines. And so we would believe that many of these neurons are likely to release GABA as well, even if they don't express VGAT. VGAT is not necessary for this process. I think this may be quite important also for human disease. This suggests, or really indicates, that the Parkinsonian state also includes a loss of GABA release. And so maybe some of the long-term hyperexcitability that is seen in the striatum in the Parkinsonian state when dopaminergic neurons die really reflects the chronic loss of GABA in that circuit, and therefore the hyperexcitability that might ensue. Lastly, neuroleptic drugs used to treat schizophrenia and many other psychiatric diseases, of course, are agonists of the D2 receptors, and they will act on dopaminergic neurons to reduce GABA release, to reduce dopamine release, but also to inhibit GABA release. And this may have important consequences for the mechanism of action and for the activity of the circuit. Okay, so that's it. Let me just give some thanks. We have excellent technical support in the lab that makes these kinds of projects possible. These are double and triple transgenic animals that are modulated with viruses and whatever else. So technicians, Caroline Johnson and Jess Solnier, helped tremendously in getting this going. Brad Lowell gave us the VGAT conditional animal and was an important collaborator in this project. Of course, Chip Gerfin, as I mentioned, and Nat Hines and the GenSat project for all the back transgenics that really make this kind of study possible. Brian Roth gave us the HM4D, Carl Diesroth the channel rhodopsin, and the double inverted orientation backbone that we now use routinely in our lab to make things like the VGAT and VMAT rescue viruses. And viruses were made mostly at UNC, and then some funding. And so this is the lab. I already pointed out along to you Nick and Jun, who did the dopamine inhibition work, R.P. Saunders and Jenya, who's unfortunately not in this picture, who did the circuit remodeling work, and Hung-Bae Kwan, who did the original cortical work last year. So, thank you. Yes.
This may be a little off the topic, but it was a great talk.
Thank you.
I have two identical twin grandchildren, boys, and they both are quite bright. They communicate well. They understand a lot, but they were very slow at learning to talk to people that only understand English. So my daughter, their mom decided to separate them more often. So their dad would take one off to one activity, the other to another, or they did the same when they were playing with other children. Then their ability to speak English started picking up quickly. So what I wondered is, one, is there any understanding on a molecular level? And two, is it too much of a leap to ask how your work might influence early learning?
Well, it, of course, is pure speculation on my part, because I have no data towards that. I think that the work can have a real influence on early learning, because we think that via these mechanisms, patterns of activity that turn on particular parts of the brain may reinforce the activity of that part of the brain. And so particularly, I'm thinking about two kinds of diseases. One is ADHD, attention deficit hyperactivity disorder, where one could speculate that some early manipulations, either pharmacological or environmental or whatever else, or stress, might lead to activation of the direct pathway in a way that reinforces itself. And what I didn't show you here is that these perturbations are actually maintained towards later in adulthood. So I think that's one possibility where that might come in. You know, the role of the basal ganglia and language and these kinds of things are still unclear, although there is evidence emerging that it does play a role there. So it could be, but I don't actually have any data, of course. On the right. My right.
Okay. Well, thank you for this very beautiful talk. I really enjoyed it. To separate intrastriatal effects versus loop effects when you knock out the GABA release in the D1 or the D2 pathway. You suggested that there's an change in firing rates in the output nuclei of the basal ganglia. Did you actually record activity in those mice?
Right.
And did you find that prediction?
Yeah, so that's a very important thing, is that I've presented this loop as if these changes in activity actually occur. I haven't shown you that they occur, and in fact I can't. So you have to remember, these are animals that are about P8 to P12. When animals are placed under anesthesia, the majority of recurrent activity in the basal ganglia, of course, is shut down, and the striatum becomes quiet. So in order to do the experiment that you suggest, we would have to record from a P8 to P12 animal that's unanesthetized and presumably engaged in some kind of task that actually turns on the striatum. And I think that's quite an impossible thing to do. So we don't know that. We, of course, do know that in adults, we've done many of these experiments, the loop acts in the way that it should act, and as proposed by these models. But we don't know that in the young animals. However, the locomotor effects make me believe that that's true, but I have no way of addressing that. The second question that you asked was about intrastriatal connectivity. And you're absolutely right that medium spiny neurons send axons within the striatum that can mediate inhibition of the striatum. As far as we can tell, the degree of innervation within the striatum of direct and indirect pathway neurons is essentially the same. They both ramify within the striatum, so it'd be hard to explain how that could explain our results. Furthermore, we went on to directly manipulate cortex and we saw the same thing. So we think it is the recurrent activity in the loop, but these are very important caveats that you're mentioning and the things that we do address more thoroughly in the paper.
Again, it may be that it's difficult to perform the experiment, but I was wondering if you have some idea if the extracellular amount of dopamine was different between those two different mice, given that that's part of the loop. And second, there's a lot of literature that suggests that interaction between the D1 NMDA receptors and D2 AMPA receptor system that could basically explain a lot of what you see with regard to glutamatergic transmission.
The first part, I accept that there may be changes in dopamine. It would be very hard to do that kind of amperometry in vivo. The second part I don't quite get. I don't really understand what exactly that alternative would be.
Well, if one of the spiny medium neurons has particular postsynaptic D1, you may think that activation could lead to the incorporation of NMDA receptors or glutamate receptors to the surface, while the ones that are enriched in D2, if activated by dopamine, are negatively coupled and would actually not promote.
Right. So I think what argues against that model is that when we inhibit the direct pathway, we see changes in both the direct and in the indirect pathway, which are occurring in the same animal in the same context and experiencing the same amount of dopamine. So I don't think it has anything to do with the postsynaptic response to the context, but rather the presynaptic manipulation. Yes.
So you mentioned some effects of an agonist of the D1 receptor on spine density and so on and so forth. What I wanted to know is whether there's a defined a window of plasticity where this occurs, or whether you would expect these effects to be true also in adults.
Yes, I think that's a great question. In cortex, what we've shown is that the ability of glutamate to induce the growth of the new spine falls off dramatically. Now that's in a sensory cortex, specifically in the barrel cortex, in which you could imagine that there should be a window of postnatal development that refines the ability of the animal to detect its sensory environment. The basal ganglia, I think, is fundamentally different, and I actually favor what you suggested, which is that plasticity will remain in that structure until much later. We haven't tested it yet, but the basal ganglia is what allows us to form new associations between sensory environment and reward throughout life, right? And that suggests, of course, that this is a plastic structure that should be responding to dopamine by manipulating its circuitry throughout life. We need to examine that. We favor that model, but we haven't shown it yet.
Thanks.
I had a question related to the nature of these loops. So in the introduction, you said that though you show a single loop, it's in reality multiple parallel loops. So what I was curious about is when you do these very broad manipulations, why is the reinforcement so specific in the outcome on certain motor behavior? And can you selectively stimulate parts of that thing to get more selective reinforcement of individual behaviors?
Right, so these are very good questions to which I really don't know the answer. The topography of the loops in striatum is fuzzy, okay? It's not like in sensory cortex in the visual system where there's really point-to-point projections. So there's a general topography there, but it's not as tight. And I think that naturally leads to some divergence in the circuit. Of course, we started with relatively gross manipulations to see an effect. These are effects, though, that were limited to dorsal and largely dorsal medial striatum. So we didn't infect the ventral striatum, the nucleus accumbens, or the olfactory tubercle, or any of those structures, and we haven't analyzed them at all. So I think there is some specificity to it. We're now trying to do what you suggest, and I think that that experiment is best done by starting in cortex with very small focal manipulations and then tracing the pathway through. And we hope to both define the scale of the loop, right, and its topography, as well as see if these effects are preserved. Okay, thank you everyone for coming.